Thank you all for joining us today, both virtually and in person. We are delighted to hear from a seasoned bioinformatics scientist with background in algorithm and software development. She has developed many tools that are widely used in population sequencing projects and medical sequencing studies, such as the 1,000 Genomes Project, the Human Genome Structural Variation Consortium, and the Alzheimer's Disease Sequencing Project. Since joining the University of Pennsylvania, her team has played a vital role in the Alzheimer's Disease Sequencing Project Structural Variant Working Group, uniquely positioning her to lead her lab in unveiling the etiology of Alzheimer's disease. Dr. Wan-Ping Lee holds the position of Research Assistant Professor in Pathology and Laboratory Medicine at the Perelman School of Medicine, University of Pennsylvania. Concurrently, she serves as the Associate Director of IT at the National Institute on Aging Genetics of Alzheimer's Disease Data Storage Site, and is the co-chair of the Alzheimer's Disease Sequencing Project Structural Variants Working Group. Please join me in welcoming Dr. Wan-Ping Lee to the podium. Thank you.
Oh, yeah. Thank you so much for the invitation, and I'm so happy to be here. And before I joined Penn, I was working in industry for 7 years. But since my postdoc training, I have been working on the whole genome sequence data since 2009. So today I would like to show you how I am using whole genome sequence data to study two diseases, Alzheimer's disease and progressive supranuclear palsy, PSP. I think disease may be not the key point today. I think the key point today, I would like to show you the opportunity and challenge of using whole genome sequence data for association analysis. But even the key point is the data, but I still like to show you so far what my understand about the Alzheimer's disease and PSP is here. For the Alzheimer's disease, you can see from the Alzheimer's brain, you can see the tau and beta amyloid in the Alzheimer's brain. And Alzheimer's, it's a mixture tau, 4R and 3R tau. And Alzheimer's disease, so far, we have most significant gene factor is ApoE. For the PSP, we only see the, tau protein for the PSP brain. And the level of 4R tau compared to the Alzheimer's disease is much higher than Alzheimer's disease. There is no 3R tau. And no, I think the 3R tau is the level is much lower. 4R tau is much higher. And the most significant genetic factor so far for the PSP FT. So that is a very brief background of these two disease. So I think let me escape from the disease and back to my conference on the data. The Alzheimer's sequencing project, if I recall correctly, it started in 2012. And in 2018, we have the first run release, R1. We have 5,000, close to the 5,000 whole genome sequence data. And for the 2020, we have the R2 release that is about 20,000 whole exome sequence. And my study is focused on the R3. We have close to the 17K whole genome sequence data. Currently, R4 is available for everybody to apply and see the data. R5, as I learned from Lisa and Jerry, I know probably, I say 2023, but I think probably this year we will have R5. That is 60,000 whole genome sequence data. Not only the genetic data, we will also have a phenotype data from the consortium, from the project, and we will have some functional genomic data from the project as well. So when we see any signal, we can always go back to check the functional data. Are they available for us to understand the functional of the variant we detect from the genomic data? And again, my research is focused on the R3. We are using 17K whole genome sequence. So the first part, you are gonna hear. I divide my presentation into 3 major sections. You are gonna first hear is the single nucleotide variant, short insertion deletion, and structural variant association analysis on ADSP R3 17K. That is the first part. If you like to know more detail, you can check the preprint on the medRxiv. The data set overview for the ADSP-R3 17K data is a multi-ethnic data. You are not surprised. The majority is non-Hispanic white, is about 62%, and we have African American is about 18%. For the Hispanic, we have 20%. Alzheimer's disease case and control portion is about 50 cases and 41% control. And if you combine these two information together, you can see for the Hispanic and African, we have more control than cases. I think that just reflect one truth is for the minority. They may hesitate go to hospital. Can I say that? Or I mean, it's not that easy to collect the sample from minority. And for the non-Hispanic white, you can see the number is opposite. We have more cases compared to control. But here on the right-hand side is all the number we have for the R317K data. And again, I mean, this is a multi-ethnic data set. So then also the data is collected from a whole cohort and sent to a multiple sequencing center. Why I like to bring this up, because when you do the association analysis, you cannot ignore one fact is that you may have a batch effect in your data set. And you cannot just adjust those covariant, because if you adjust the covariant, just simply adjust those as covariant, your signal will be gone. I mean, the true signal will be gone. So here, just like to bring one idea up is that when you are working for a consortium data, that is quite easy. You can see, data sequenced by different center, data sequenced by different machine, data sequenced by different library. Even the read length, whole genome sequence data read length is different. And everything here you see gonna be your batch effect. Then how are you carefully to to make sure your analysis is in the right direction? Okay, so the multi-ethnic data, then the first one we will do is do the principal component analysis. If you remember, I think a long time ago when a talent scientist is perform principal component analysis on the European genomic data, then you just shift the angle a bit, then you can see the European map. So that is how amazing the genomic data is. You really can tell, the geography map. And the same here, but not that ideally. The same idea here is that when we do the principal component analysis on our genomic data, we can aggregate here. I believe is non-Hispanic white, and the blue one curls from the non-Hispanic white to the Africa America is what we call Hispanic. Okay, so that is our data set. And you would like to make sure our data set reflect the the same truth from other projects like 1000 Genome project or Human Genome Diversity project. Those two projects, they have, for example, for the 1000 Genome project, they have 26 population, and you can aggregate them into 5 continental group, which is East Asia, and here is the South Asia, and you can group them into European, that is the green here, and here is the African population, and across in the middle is American population. So when you combine your data with 1000 genomic data, you should see your clustering, should close, maybe not identical, but should close to the 1000 genome data. So for our data, here is African American. That is the same as African in the 1000 genome, and Hispanic is talking about the American in the 1000 genome data. And European is the blue one here, so that is the same as the European data in the 1000 genome project. So then you can make sure nothing wrong in our data. And for the human genome diversity, you just do the same practice to show your genomic data. Nothing major wrong compared to their data set. So that is the first step we will do the principal component analysis. Okay, then I think recently we start to talk about how reliable self-report ethnicity is. That when we have the data, when we collect the data, we will ask, are you identify yourself Hispanic or non-Hispanic? And do you think your ancestry tree is European, African, or Asian? So from our data set, we have African American, and we have Hispanic, or we have non-Hispanic white, and we have others. We don't know the answer. They don't answer this question. So now, based on their genomic data, how much their genomic data to their answer about their self-report ethnicity. From this analysis, we find out that it's about 92% match to each other. Some difference would be people, they identify themselves as African American, but we see from their genome, it seems like more African. Or some, they will say they are Hispanic. Hispanic could be, I think this one could be, most of them, it's how to say, if you look at the Hispanic population, you will see the majority of their genome is from European, it's about 70%. But some of them, they claim Hispanic, but when we look at their genomic data, is African American. It's about 600 in our data set. But most of them are matched. When they say Hispanic, then from the genomic data, we see Latin American. And non-Hispanic white, then you can see from the genomic data, we class them into European ancestry. So overall, our between the self-report and genetic infer ethnicity is about 92% match rate. It's high. Okay, so what we do is that we aggregate all the sample together, all the sample we have together to perform association analysis, or we break them down into the subgroup for the non-Hispanic white, African American, and Hispanic. The reason why we like to aggregate them together because we like to increase the sample size. Also, we like to see, is there any common factor across the population contribute to Alzheimer's disease. Okay, so that is the reason why we pull all the sample together. The reason why we break them down into different ethnicity, because we like to see is there any specific population genetic factor contribute to Alzheimer's disease. So we have everything together. We also have break them down into the population subgroup, and we have done the common single nucleotide variant analysis or rare aggregate analysis, because this is the whole genome sequence data, so we can work on the rare analysis. For the rare aggregate analysis, we done it on the coding region and non-coding region. So let me show you the result. You will also see, I mean, as I mentioned, you will see the common single variant analysis. You will see the rare aggregate analysis. And I will also show you my favorite topic, that is the fourth signal you can see from whole genome sequence data. And I will also show you our result of the structural variant and copy number variant. The first one is the single variant analysis. We apply this analysis on all the common variant that is minor allele frequency larger than 0.5%. We don't have too much more. All the ApoE signal, I mean, we already know ApoE is there, and we also know the CR1 and BIN1. The new one is, occur on the African American sample that is in the chromosome 21 link code. Here is the long RNA gene region, and we don't have another data set to validate this one. So if you happen to know there is some other data set we can validate this one, then that would be good. We do this analysis on the single nucleotide variant, also show insertion and deletion. And again, I think those signal, we already know them before. For the Hispanic group, this spike is for the early onset Alzheimer's disease. I will show you more information about this one. ApoE as the most significant variant, we break them down into the to check the allele frequency between cases and control. E4 as a risk for Alzheimer's. So you can see allele frequency for the E4 for the cases is always higher than allele frequency in the control. And I also show you the allele frequency from UK Biobank as the control number here. And the E2 as a protective for Alzheimer's. You can see the E2 allele frequency for cases is opposite. It's always lower than allele frequency in the control. And on the right-hand side is the odds ratio breakdown by different ethnicity for E2, for E4 and E2. And E4 as a risk factor, we can see homozygous E4 may aggregate on the cases. So we like to see, is that true? Then that is true. You can see for the African American group, for the E4, we have 15% homozygous E4. For the control, only 2% homozygous E4. Then you may ask, I mean, are those 2%, they are not yet developed Alzheimer's or some other protective factor for them that we don't know. And the Hispanic, the homozygous ratio between cases and control is 6 to 1%. For the non-Hispanic white, the homozygous ratio between cases and control is 7 to 3%. So that is what we see from the data. And I think those are knowledge we already know from the previous study. As I just mentioned, there is another peak for the Hispanic cohort. What is that peak? And I say that is for the early onset Alzheimer's. From our data set, we see some of them since I think their onset age is quite young, and we see that it's a peak. Let's go back to the Manhattan plot. That is a peak here, and this peak is all linked to a mutation called G2068 in the precinct 1. We have 17 G2068 carrier in the ADSPR3 data, and they are all self-report as a Hispanic subgroup. And this region, I think we already have, several study on this region. If you are G2068 carrier, then you will have a much earlier onset age of Alzheimer's. That is 58.6 years old, compared to if you are not a carrier, it's gonna be 74.7 years old. And you can see, that is quite different between these two onset age. So if you are carrier, then you will have such huge risk to have early onset Alzheimer's. Okay, so we see this. Then one question we like to ask is that even we say that it's Hispanic, but Hispanic where, from which region? So again, we use 1,000 Genome Project data and apply the principal component analysis again. Then we those carrier seems close to the Puerto Rican, and that I think that all the previous study also show this mutation seems only detect in the Puerto Rican area. If that is in the Puerto Rican area, then we also know the Hispanic group. If you look at the entire genome, about 70% of their genome is European. Then why this mutation only happened in the Puerto Rican? So local and sensitive analysis. Again, for the Hispanic group, for those carrier, if you look at genome-wide, it's about 70%, they are European. But if you look at this region, if they are carrier and the solid dot, meaning they have this mutation, you can find the solid dot as always happening on the African ancestry genome. Everyone is following this principle. So that means, I think this mutation is either, somebody, when they move from African to Puerto Rican, then evolve this mutation, or somebody got this mutation and moved to this area and somehow evolve other mutation and locally and this one become the risk mutation to Alzheimer's disease. But this mutation, again, is only happened in the Puerto Rican sample in our data. So from the global and local and sensitive analysis, you will get idea this region is inherited from where. Then you can trace back the reason why people, they have this mutation and what contribute to the disease. Okay, so nothing special for the rare coding and non-coding analysis. We just find ABCA7 in the coding region seems significantly associated with Alzheimer's in the non-Hispanic white group. And for the non-coding rare aggregate analysis, we find that TOM40 on the chromosome 19, very close to ApoE, solely contribute to the Alzheimer's when we use the pool sample everything together analysis. Then when we break down into the African American and non-Hispanic white analysis, they are subjective. Significant, but they are not significant for the Hispanic group. So nothing special for this rare aggregate variant analysis. And okay, so now let's move on to my favorite topic, that is the false signal. We see there is, SMV in the ANK3, that is here, chromosome 10. And this one, I think, when you check the literature, you also can see this one seems, somehow linked to the neurodegenerative disease. So we are quite excited about this new signal, but unfortunately, this one is not true. I will show you the reason why this one is not true. And we also detect another 12 short insertion and deletion. They are significantly associated with Alzheimer's. Again, we are also quite excited about this finding, but the validation from Dr. Jerry Schoenberg's lab showing that none of them is true. Then I will also show you the reason why none of them is true. For the first one, this signal from ANK3, you can see it's a SNP and very close to each other. And I will say nothing wrong with this one. The only something wrong is that they are too close to each other. When you look at the whole genome sequence data, you will expect to see one SNP in 1,000 base pair region. But this one, then you will start to sense maybe something wrong for the artificial alignment. If you go deeper to check the alignment, you will see the alignment quality of those regions is very bad. And somehow the tool using those bad alignment for variant coding, then we see this one. I'm not 100% those are false positive, but according to the alignment quality is very low. And I think I don't have that confidence to report this one. Low quality alignment, also they are too close to each other. So I think I don't believe they are true. And the next example is the false indel. That this one is also the coming from the artificial alignment. If we use HG38 as our reference genome, and from the sample you can see here is indel, short insertion and deletion. But we also see between two reference, HG38 and T2T, they don't concordant to each other. And just because the reference issue, we see this indel popping out, and then the PCR validation, I think none of them we are able to validate. So in another word, when you see the short insertion and deletion, that would be better for you to check another reference. This reference T2T telomere to telomere is a complete reference. Then this T2T reference may give you the different answer. This one is also quite interesting. You can see, I mean, SNP aggregate in this region, and when we extract the sequence inside this region and then do the blast with other species, we find out this may be a contamination because the sequence here we can match with bacteria 100%. And for that bacteria, there is no SNP in that bacteria. Just this region is quite similar to the bacteria region. So you will see those SNP popping out aggregate in this region and somehow significantly associated with Alzheimer's. You will ask, I mean, if that is contamination, that should be randomly distributed to all the sample. Why it becomes significantly? I cannot answer that question either, but sometimes you will just see error or doesn't spread randomly. So when you check your results, just be careful about this could happen just because this is a contamination of bacteria in your sample. This one is also quite interesting. HG38, GRCH38 for the chromosome Y is incomplete. So if you are using this human reference genome, you will see some SNP is male only. I mean, nothing wrong for some SNP is male only. But when you see this one, you need to, get a good sense. Is that because the incomplete of chromosome Y? So when you process your data, you cannot map this read back to chromosome Y. And instead, you just aggregate those read to a very similar similar sequence region with the chromosome Y. And because that is not a chromosome Y, so you will start to see SNP in this region. But the truth is that short read actually is coming from the chromosome Y rather than other chromosome. So example here is that on the chromosome 3, we also see this one significantly associated with Alzheimer's disease. And this is male only. For using HG38, we don't have any female having this SNP. But using HG38, you see male, they have heterozygous SNP for this region. But if you move to using T2T, you can see, I mean, we don't have this SNP. And for the T2T, we don't have this SNP. For those male, they have SNP against using HG38. Why? Because T2T is complete genome. So now using T2T, you are able correctly map those short read back to the chromosome Y. And for the chromosome Y, there is no such SNP popping out. But if you are using HG38, you just map those reads, I mean, in a similarly, but not 100% identical to the chromosome Y region. You just map to a, very high similarity region, and it's not 100% the same. Then you will see this SNP popping out. So again, if you see the male-only variant, then just be cautious about your finding. Okay, then the structural variant, we also detect structural variant of deletion, insertion, duplication, inversion, and translocation. Same as the SNP, we find that African American sample, they have a higher number of structural variant detect. And we also, again, using the PC to look at our data. The left-hand side is the PC analysis based on SNP. The right-hand side is PC analysis based on the structural variant. You can see they quite concordant to each other. Then that is the evidence showing that our result is fine. But if I tell you the PC3, the third principal component, is linked to the sequencing length, then you will sense how hard to detect a structural variant, because structural variant it's quite affected by your sequencing library or your sequencing center. But for the PC1 and 2, you can see between SNP and structural variant, they quite match to each other. And when we done structural variant calling, then we start to analyze, is there any structural variant located in the LD region with the non-SNP, with the non-Alzheimer's SNP. So this is a quite interesting finding, is that this two SNP is a new novel SNP associated with Alzheimer's disease. Then we see there is a deletion in the promoter region of NCK2, and the R square, it's 99% along with those SNP. So maybe the true impact of this SNP is from this deletion located in the promoter NCK2. So nowadays, if you are studying on the structural variant, I mean, the first step will be you may like to check, is there any non-existing disease risk sitting in the region with your structural variant. This table is, I only copy the first half of this table. If you go to that preprint, you will see the full information. We also do the structural variant association with Alzheimer's disease. These two duplication I highlight because I am able to replicate these two duplication on another data set, on the R4 data set. Okay, so especially for this RAB-GAP1 98 base pair duplication, is a rare duplication, and it show as ratio is 0.86. It show us a protective factor. Then when I check the cerebrospinal or floor amyloid beta level, I find out for the control group, if you are carrier, then you will have much lower amyloid beta level if you are carrier, again, because this one is a protective factor. If you are non-carrier, then your level will higher. And for the memory score, if you are patient and you are carrier, your memory score will be lower compared to the non-carrier. That may imply that if you are carrier, then your decline speed will be much faster. I don't know the reason why, but that is just what we see from the data. Okay, so I think I stop here. I show you how to use the whole genome sequence data for the analysis, and how important the population diversity is in your analysis, because you need to check the population factor. And whole genome sequence data enable short insertion, deletion, structural variant, and rare variant analysis. But you need to be super cautious about the false signal from whole genome sequence data. For the second part will be just very quick. The paper preprint is here again, and this study that Alzheimer's Forum is also noticed our this study. So we have a short post on the arts forum. So you can check it out if you are interested. Manhattan plot here. The black one is the non-PSP loci, and number one is the new PSP loci. Everything here is European ancestors tree because there is nowhere we can collect other ancestors tree for PSP study. Something interesting is that for the E4, if you still remember, E4 is a risk factor for Alzheimer's disease, and E2 is a protective factor for Alzheimer's disease. But on the PSP study, it's opposite. E2 become a risk. So you can see the allele frequency of the E2 in cases group is higher than the control. And E4 on the opposite, again, showing as a protective factor for the PSP. So I think that is quite interesting because, I mean, there are some similarity between these two diseases, but why this, I mean, significant genetic factor for Alzheimer's showing the different direction, impact direction on PSP. We also done the structural variant analysis, and not surprised, we find a MAPT. And we also try to, because MAPT is just a gene, but for this famous region for PSP, they have some bigger structural form. So we like to understand, I mean, is there any other structural form contribute to the disease? The disease is PSP. So we focus on the 3 duplication, alpha, beta, and gamma. The size of duplication is here, and here is their coordinate. We like to ask, do alpha, beta, gamma contribute to PSP independently from H1 and H2. H1 and H2 is the non-risk, non-genetic risk for PSP. So that is the first question we like to ask. The second question is that, is there any combination of alpha, beta, gamma contribute to the risk of PSP? Or is there any structural form alpha, beta, gamma associated with the non-MAPT subhaplotype, such as H1C, that is a famous risk factor of the PSP? The answer for the first one is yes. When we have more copy of the gamma, our odds ratio will increase. And we also see there is a structural form link to the H1C, the famous risk of PSP. So from this copy number variant study, we have those conclusions. Okay, so here, interestingly, is that I showed you the E2, the E2 factor for the Alzheimer's and PSP, they have opposite direction. And copy number of the gamma contribute to the risk of PSP independently from H1 and H2. And of course, we need more sample to validate our finding, and we need more sample from other population. We are working on that. Okay, so the next part, I think I would just like to show you when you have a whole genome sequence data. And because whole genome sequence data is still more expensive than genotype array data. So how can we integrate them together and reduce the cost? The pipeline we think is that we probably can propagate our knowledge of the whole genome sequence data through imputation panel construction. And then we can impute our genotype data. When we say imputation, then you will argue TopMan, they have more sample, so they will have a better quality than us. So how can we overcome this disadvantage of our data with the smaller data set that we can from the bioinformatics tool, using this bioinformatics tool to combine two imputation together. So you will have imputation to show the disease specific variant. Then you can also gain the advantage using the panel having a bigger sample size. So that is the way how we like to integrate between whole genome sequence data and genotype array. I think I will skip the result here. And the summary of this one is that, as I keep saying, that we are thinking how to propagate the knowledge we learn from whole genome sequence data to the genotype array data, because genotype array data is still cheaper, and then it's more available to access them compared to the whole genome sequence data. For this study, of course, we already have this pipeline, but we see there are some drawback of the existing tool. So we need to develop a new tool and best practice to construct a disease specific reference panel. Okay, so I would like to wrap my presentation. Thank you, everybody here. I don't have enough space to show everybody's name, but quite thanks to two of my postdoc, Hui Wang and Bo Liang Zheng. Bo Liang Zheng is here, and also thanks to two of my mentor, Li Sang Wang and Jerry. They are also here, and of course, thanks to the Timothy Chen in the UCLA, teaching me a lot of knowledge of the PSP study. And this data set, again, it's contributed by so many cohort and working together. So that is a quite large consortium, and like to thank everybody working on this collaboration and project. Thank you. I will stop here.
Go ahead and take some questions here and then also online. So those online, you can unmute yourself and ask your question if you have one.
Maybe I can ask a question. So Wan-Ping, this is it's great to see how complicated like whole genome analysis and how it's like false positives, like so on. So it's great. One question I have, which you and I talked about, is there are two new reference genomes being introduced. One is the T2T and the other is the Pan genome. And we talked about like how this is going to change how we do data production. Maybe you can comment a little bit about what is your take on these two reference genomes, and what's the occasion you should use one and the other?
Okay, yeah. I think HG38 was released in 2013.
13.
Yeah, it's a long time ago. I think 10 years already, but we are still using that. Even sometimes we will use the one version back to the 30. And the T2T, it's a complete genome. You can tell from the name is telomere to telomere. The approach, because I don't have that bandwidth to reprocess all the sample with the new reference genome. So I usually just cross the check region. The region of the interest. I don't have that bandwidth to reprocess everything. So if you find something interesting from HG38, then maybe you would like to check the status of the T2T. The second reference genome Lisa mentioned is the Pan genome. That it try to describe the variant across different population. Either HG38 or T2T, if you think them in a graph, it's like, I mean, one point to another point, it's like a line. For the Pan genome, now you can create different branch, different paths to describe the variation of different population. So from 1D to 2D to two-dimension graph, you can see the computer science complexity increase a lot. So again, I don't have that bandwidth to reprocess everything, especially for the Pan genome is premature. But I will encourage everybody, if you see something interesting, maybe you really need to, I mean, go cross by different region and checking are they consistent to each other. If they are, then I think your confidence will be increased because there may be no reference bias.
Dr. Rader, you can go ahead and ask your question.
Yeah, hi, it's Dan Rader. I'm online. Thank you so much. That was really, really interesting talk. I had a couple questions about your PSP GWAS. It's so interesting. One is I was just kind of blown away by the ApoE2, ApoE4 associations. And I guess one question I had is this the first sort of neurologic condition in which ApoE2 has been found by GWAS to be a risk allele and ApoE4 to be a protective allele? And then maybe I'll just, and my other question is, given the kind of the phenotypic overlap between PSP and Parkinson's, and the pathologic overlap between PSP and Alzheimer's, including involving tau, it went kind of fast, but I'm just wondering if you could go back to your primary GWAS. And I'm interested in if there's overlap of loci with PSP with either Parkinson's or Alzheimer's, or all the loci other than ApoE, like completely new loci.
I see. For the E4 and E2 opposite direction for between Alzheimer's and PSP, Dr. Schoenberg, they have a paper back to the 2011. It's not significant, but if you look at their allele frequency, you can sense, I mean, they also show maybe the impact of these two alleles are different from Alzheimer's disease.
Was that PSP also? I'm sorry. Was that PSP?
PSP.
Okay.
Yeah,
And
PSP
earlier GWAS of PSP, right? Got it.
Yes. Yeah.
Yeah.
And between 2011 and to now, of course, I think the number we see here is really quite significant, but we cannot ignore that maybe some sample selection bias in our study. But between this, I mean, decade, you can see, I mean, there are some other study, none of them, I mean, really claim, but all of them just, I mean, imply saying these two alleles may show the different impact between Alzheimer's and PSP. Okay, then back to the common variant between these two disease. We are working on
Can you
that.
show us your Manhattan plot while you answer the question? Yeah. Yeah.
Yep. So looking at this one, only ApoE and MAPT, I think some previous literature may say they contribute to Alzheimer's disease. Others, I think, seems belong to PSP specifically. Yep.
And there's one other disease where it flipped like in PSP, and that's AMD. Is that?
Yeah, eye disease.
It's an eye disease. Yeah.
Yeah.
That's the only other time that you see this
Yes.
reversal.
Yep.
really fascinating. Thank you.
Any other
I have
questions?
one question. I'm Yuanqing from a research associate from Tesco Lab. Really nice talk. I also recently analyzed some GWAS data set. I want to ask a technical question about the PC numbers. I see you using 5 PCs to correct the population stratification. Also, you also mentioned that if you correct too much, the signal will disappear. So how do you determine the number of PCs used for the GWAS analysis in this study?
Oh, yeah. I see. I think for the PC, let's go back. I think as my first the first run analysis, I was just using very very little covariant, maybe nothing, and then first run I try, and then I will start to add covariant one by one from PC1 and then to PC10. Of course, from my neural model, I will already know which one contribute to my analysis. And I think you are talking about this one. Should I adjust sequencing center or not?
Yeah.
I didn't adjust sequencing center because some sequencing center, I think they only sequence cases, or they only sequence control. So if I do that, then that will be quite how to say, then I think I may miss that true signal if all my cases just sent to one center, all my control sent to another center. Then if I just sequencing center, then I think the signal will be gonna be gone in my analysis. That is another example here. I mean, everything is quite balanced. I mean, between case and control. But again, I think from my first run analysis, I will just use a very little covariant and then start to sense which covariant contribute to the association analysis. And then once I have a signal, I will go back to check. Is that signal, specifically belong to any sequencing center? If that is the case, then that one could be a batch effect. Yep.
Okay, thank you.
A quick follow-up to that, what is like, do you see a lot of inflation on your Manhattan file? That might be a good way to check if that's a serious.
Yes, yeah.
Yes or no?
Yes. So you will see. I don't remember which one when I adjust, then even the ApoE will be gone. So then I think you will know. Okay, so that is not right to adjust that.
I remember your Manhattan plot looks pretty like
Yes.
calm, right? A lot of inflations.
It's pretty calm because ApoE, built something like a tower. So others, it's it's pretty calm. But if I check the QQ plot, it's look normal. Yeah,
It's good.
Oh, we have a few in. Go ahead.
Yeah, so I wanted to ask about CRISPR-1. So you have a variant for CRISPR-1, right? And then you're saying it's dominant in the Puerto Rican subgroup and it's coming mostly from the African ancestry, right?
Yeah.
But you also have a GWAS with African Americans, which is that same variant present in there, or they're from different regions of Africa? And then also CRISPR-1, because it's also part of familial, like Alzheimer's disease. So I know like there's some studies also in Cape Town, for example,
Yeah.
on that.
So-
So what's going on there?
Yeah, so let's go back to the Manhattan plot. You can see that is not a single variant. It's a bunch of other variant, right? But if you look at this one specifically, that only happened the Hispanic, Puerto Rican. For the others, I check the allele frequency on African American. Their allele frequency, some of them is higher than Hispanic group. meaning that they indeed having other mutation, but this mutation only happen in the Puerto Rican area. So again, either someone very special from African carry that then to the Puerto Rican. Yeah, and yeah, but I cannot answer that. Yeah, that question. Just from the data, yes, I think for other variants, you can see the high allele frequency in African American. But this one specifically only for the Hispanic Puerto Rican.
So the local ancestry is African, but this specific variant is specifically Puerto Rican. Is that what you're saying?
Yes.
Okay. That's interesting.
All right, you had a question.
Hi, Dr. Lee. Thank you so much for your wonderful presentation. I had a couple of questions, but the one I want to start with is I thought it was really exciting how you shared you're going to impute the genotypes and try to merge them with the genome data. I was wondering what your thoughts were with regards to imputing the exome data so that you can keep the rare variants. There's been, people doing different combinations to see how much information you can get from various combinations. Is that something that you've looked into or something you would consider, especially if you have exomes on, a bunch of individuals who are unique from?
Yeah, nice suggestion, because I think I'm busy enough for the whole genome sequence data, so I don't think about how to handle the whole exome. But I think we could try to impute that once we have that panel, and the panel, the input for the panel is VCF. So you can just input the VCF either from the genotype data or either from whole exome data. I could try, as you say, because I have whole exome data, why not doing that? Yes, but I haven't done.
And if I can, I just want to ask one more question. Right at the beginning of your talk, it was very interesting also to see that there were E4, carriers who were controls. Are any of those like super old controls, and have you done any candidate gene analyses? I'm particularly interested in any rare variants in Relin or others as well.
Yeah. I think then you may say for this 2%, for example, for this 2%, maybe they are still young. So they haven't developed Alzheimer's disease. I didn't go that deeper, but I think I can do that. Then for those 15%, are they super old or not? Again, because I didn't go that deeper, so I don't know the answer for that one. Yeah.
The controls are probably a decent age. You wouldn't expect them to be that young.
Yeah.
But that's super exciting.
Yeah.
Thank you so much. The discussion have other places to go. Thank you so much, and we can continue the communication. So we can share your slide online, right?
Yeah.
Okay. Yes.
Yeah, I will
Post
send you
it
the copy.
online on the Center for Global Genomics and Health Equity channel, so that all those you can share with your other colleagues. Thank you so much.
Thank you.